So, good morning, students. So, I am very much fortunate of having this opportunity to present how to make your research paper e-poster. Actually, what we do in MSc level or let's say in BSc level, we do lots of research, we do lots of experiment. But the thing is that should be compiled in a very proper manner so that in future perspective you can just uh, what you can do, you can just present your that poster to any of the conference and you will get benefited of it. Actually, uh, participation in the conference is a good thing, but participation along with your research paper, your research work, or let's say your poster presentation is a way better. So here I will try to share some basic concept or what exactly the poster will contain that we have to see so that if you do MSc in future, let's say in any of the life science stream, so there will be a common one project will be there, minor project, so that you can draw some papers as well as poster from that. So I will share one slide that is very basic one, but as you should know what are the criterias how to compile our so much research work, because the research paper will be having, let's say, uh, 3 4 or let's say more than 5 pages also. So how to gather those information into a one slide and making those as a e-poster, that we have to see why we are doing so. Because nowadays, due to, as madam told, like due to pandemic situation, most of the conference are organizing on online platform, let's say Zoom or something. So it is way more convenient for us to present e-poster on those conferences. Moreover, what you can do, you can you can share your e-poster in that conference, and they will be having some best presentation award, and you can grab those presentation also. So we will start with some basic one. So I will share one slide. So this is the simplest slide one can have. Okay. So what you can see here is nothing but the title of your research study. It is visible.
Yes, it is visible, and I request all the students to zoom it and see it so that you can see what is written over here. You can zoom it on your mobile phone.
Okay. So at very first, what we have to write is our research title. On what experiment you did your research, on what topic you did your research, that we have to see. Okay. Let's assume there is one phytochemical analysis study of any of the plant root extract or let's say leaf extract. So your title will be phytochemical analysis of that particular plant root, dried root extract from particular solvent. So that will be your research title. What those figures denote, this one, this one is nothing but it. If we talk about some institute, so they will be having there some logo. So you need to put that logo here. Okay, you can put logo here or you can put logo here. But if your project is government funded, okay, so you need to you need to put your college logo as well as the project funded by which, under which project you have to put that also. Like let's say it is DST funded or SCRB funded, so you need to put that logo here so that it will be very convenient for viewers to see the title as well as to what research organization it is collaborated with, you will that viewers will come to know very easily. Let's say, for example, this is nothing but our uh, college logo, and if it is funded by DST, then DST funded, DST logo will be there. Okay, just below it there should be a author's name or the people who did the particular research study. Okay, so let's say two of you or friends, okay, they did some research work, so both names should come there. If it is three, in collaboration with three peoples, then three authors will be there. Okay, now the thing is, the first author, there is first author, second author. So what you need to do is, let's say, uh, first author name is some sir. Okay, some sir. So what you need to do, you need to do put a- us one here at upper script. Okay, what you can do, see, all of you can see sir. Okay, and at the top there is one. That means the purely work is done, major part of this research work is done by this sir. Okay, and there will be co-authors. This is the first author, and for co-author you can put two, three and something. Okay, for second author you can put two over there. Okay, and along with those authors name, to which institute they are working in, you need to mention that also. Okay, after this presentation I will share one slide. Okay, so you will get a confirm clear idea about how research poster can be made. Okay, now this, we had ended up with the title of the research paper and the authors. Authors will be just below the title, so that the viewer will come to know the title of the paper as well as the author who did this research work. Now moving to the next one is nothing but the abstract. What exactly abstract is? Okay, what exactly the abstract is? Abstract is nothing but the whole summary of your research. Okay, that will contain your introduction, that will contain your factual. Okay, but that should be in very short manner, that should be compiled in very short manner. Okay, so you need to put all those stuffs in the extract. Okay, in abstract what you will do, you will put all those information but in very compressed manner, so that the main highlighted point will be get covered from that research paper. Now now there is nothing but the next section is introduction. Now introduction means what? You need to introduce what is phyto, if you are talking about the phytochemical, then what that phytochemicals are, what your plant advantages are. Okay, and what you can do in for future perspective, how it is beneficial for a human mankind. That what all you need to put in the introduction. Okay, so as compared to abstract, introduction may be 100 words more. It can be easy to put some 100 words more in introduction, as we need to introduce what we are, what we are, we have to do, what are the scope of this particular experiment, and how beneficial is that experiment for human mankind. So for every experiment, okay, you will be needing some material, uh, you will be needing some methods. So the next column will be materials and method. Whatever material you have used, whatever method you have used, you need to put all those methods and materials over this column, okay, over this box. Now, after that, that materials and method may contain some table form, okay. If it is phytochemical analysis, we are just focusing on phytochemical analysis because next slide we will be seeing some phytochemical analysis slide only. So for phytochemical analysis, there are series of phytochemicals in a particular plant. So we need to identify all those. Okay, so there are some tables like from this test, if it is giving yellow color, then it is positive. So that much methods we can't write. Okay, so the best way is just put those all information in a tabular form. So it will be very easy for reviewer also to read the uh, particular method, how they had isolated or how they had estimated the particular phytochemicals. After materials and method, you will drag down to the result. So the most important thing is you can put your result in a graphical form. That is the best representation. Okay, if you are let's say if you have some groundnuts uh, variety, okay, some six variety you have, and you need to calculate the percentage of oil in it, okay, so you just put those in graphical form so that one can easily compare that variety 1, variety 2, how much percent of oil they have as compared to standard one. So in biochemical reactions, in biochemical practicals also, you can do the same thing. Why I'm focusing on graphical presentation? Because it will be very convenient and it will be very much easier for a viewer, or let's say some whoever will read this poster, for them it will be very easier. So how to put graphs and all? You just go to the insert option. Okay, you just go to insert option. After that, you have chart option here. Okay, just click on it. You will be having some line, some columns. If I choose some line, okay, let's say some line, and here you can see various lines you can draw, but the most specific is nothing but this one or that this one. Okay, so let's say, let's click on OK. Okay, you will be open up with this Excel sheet. This is nothing but the value you got. Okay, just, and for variety, okay, just we had talked about the groundnut variety. Just say uh, groundnut, okay, GV groundnut variety 1. Okay, second series will be groundnut variety 2. Third series will be groundnut variety 3. Okay, you had put some values here. Okay, let's say this is for protein. Okay, this is for oil content. Okay, this is for fat content, and uh, this is for carb. Okay, carbohydrate.
Sir, we are not able to see a full slide.
Actually, ma'am, I'm working on, so it will be visible for to you. Okay. uh,
Okay.
okay, so I will close this one. So you can see one chart came here. Okay, it
Yeah.
is visible.
Yeah. Yeah. Yes, sir.
So you can clearly see the this one, the blue one is for groundnut variety 1. Okay, this is blue background, that's why you are not able to see. But just assume, okay, just see red one. Okay, this is for groundnut variety 2. So you can see protein content is near about 2. Okay, this value will represent your protein content. Okay, either it will be gram or in microgram. Okay, so you can clearly see protein content is nearly 2, and oil content for groundnut variety 2 is nearly about 4.5 something. So you can compile your result in this graphical form also. Is it clear? Same uh, we can compare those three varieties on this protein parameter, oil content parameter, fat content parameter, and also carbohydrate parameter. So such kind of result you should put in your research poster. Okay. Now, oh, sorry. Now, just coming to the next one is nothing but you had compiled your result. Okay. After that, what you need to do is you have to discuss your result. Okay. You have to discuss your result from earlier findings. If in case some variety will be having low oil content, and whatever variety you have used is having much more oil content, then everyone knows that much more higher percentage of oil in particular groundnut will be very much beneficial, for a further process. So that result came into positive manner, in positive way. So you can discuss here, in earlier experiment they had used some variety that will be having some oil content very low, but my finding having much more quantity, much more percentage of oil, hence it is beneficial for human welfare, or we can do further research on it. That will be your discussion, and what you can conclude from your research work will be written here. Okay.
झालंच नाही कापड करतो म्हणे ना आता.
Now, the very most important thing is putting your references. Don't skip to put your references in your research poster. References should not be in more quantity. Either 3 or 4 will be sufficient, because when you write this introduction there will be one line, or let's say there will be some statement which will be exactly same from any research paper. So you need to give credit to that particular researcher, and you need to put that researcher name here, so that it will be very much legal for you. Okay, no one can say that it is illegal. So putting references is very much important, and at the on this basis also there will be judging. Okay, if you put references, that is much more valuable. That will add points if you are presenting your presentation. So all of you clear with this?
Okay, I request all the students to keep your mic on, and then you can ask questions to Sadavarti sir. If anybody has got questions in their mind, how to make e-poster, if you have any queries, you can ask to Pranay Sadavarti sir. Anybody? Anybody has got queries? You can ask. Who are there?
This new
Oh,
poster is visible for you?
Yes, sir.
yeah,
So
there.
just see, what I had written over here is qualitative analysis of phytochemicals and antibacterial studies of Solanum nigrum roots. Here is nothing but the author's name. Okay. Now just moving to the downward, there is a abstract. Okay. Abstract as compared to introduction, it is much more smaller. Okay, but the main points of the particular research paper had been discussed in your abstract itself. Okay, after that it will be followed with the introduction part. Okay, whatever the findings, see, as I said, this Jay, okay, Jay 1968, I had been mentioned here because I had taken a particular line from his paper. That's why I had mentioned in references his name. So no one can put question on it. So this is a small tip you should follow when you are making your e-poster. After that, materials and methods. Okay, and as I told you, they if in phytochemical analysis, okay, you will be doing so much analysis for saponins, resin, lactone, cardiac glycoside, and so on. So what you need to do, put one table, okay, that will be much more easier for viewers. Name of the test, okay, you can zoom it and see name of the test, okay, observe changes and remark, or let's say result, it is present or it is absent. So you can put positive sign for presence and negative sign for absence. After that, for antibacterial activity also, you can put table. Putting your result in tabular form or in graphical form or in chart form is much way easier for viewers also, and as for you also. Okay, now just switching to the result, and result we completed here, it is nothing but the discussion. Okay, that will be followed by our conclusion, and lastly I had put 5 references over here because some of the references are from these steps, okay, phytochemical analysis steps, so you need to give credit to them also. It is okay. If you have any question, please go on. Please ask. Your questions are welcome.
Yes, anybody has got any question? Please ask. Okay. Thank you so much, sir. And I request Akash Singh sir to continue the workshop.
Thank you, ma'am.
I will make you co-host, sir. Just
Okay,
a
ma'am.
minute. Yes, you are co-host now, sir.
Okay,
Please continue.
I will just share my screen.
Yeah.
Okay, is my screen visible to everyone? Is my screen visible, ma'am?
Yes, sir.
Okay, thank you.
Everybody, please respond to the questions if sir ask or ma'am ask. Please respond. Please give your response, and you can zoom it and see it.
Okay.
Okay.
So thank
Yeah.
you, Pranay sir. That was a very good explanation. So Pranay sir has explained in quite a lot of details about how a research article has to be converted into a poster and then has to be represented. The most important part of a poster is that it is represented in conferences where people are generally walking through the corridors, and the poster should look interesting enough and it should be concise enough so that all the information is put into a single piece of paper and everything is displayed so that in one sitting or in one go, a normal passerby or a normal onlooker can have a look at it. So we will just be looking at something that has already been printed, something that has already been published by some other authors, some other researchers, and we'll just look at how concisely they have put the results and the abstracts. So this is a paper that has been created by somebody at Texas University. Texas is in USA. So there are 3 researchers. As you can see, again, reinforcing the points that have been put by Pranay sir, look at the title of the whole paper that has been given over here. It has been given a huge font so that anybody can look at it from a distance. The title says reactivation of E. coli in water after UV and photocatalytic disinfection. This is something that can be read from a distance, and it is so easy to understand कि आप यहाँ पे इसको आसानी से एक लाइन में पढ़ के you can really understand what they are trying to say. So the paper is basically about deactivation of E. coli, and there has been certain cases where after the deactivation of E. coli, after UV treatment, they get reactivated. That is, they start growing again. So the whole paper basically focuses on how are bacteria killed after UV treatment and how some bacteria basically survive and how can they be killed again. Just below the name, just below the actual title of the paper, the three authors, their name is mentioned, and then the department where they have worked has been mentioned, and then finally the email ID of the author, the corresponding author, has also been mentioned over here. So we'll quickly look at what this paper means and what they're trying to say. As an introduction, at the left, there are two images and a small explanation has been given. I'll just brief you about what they're trying to say. Everybody is aware of ultraviolet light and its uses as a disinfection agent. Okay, so even in your aqua guards, even in your reverse osmosis filters, and in generally inside the laboratories also, we have a tendency of using ultraviolet light to disinfect the area. Disinfection basically means killing of all the microorganisms that are present in that area. How does ultraviolet light do that? Ultraviolet light basically damages the DNA of the bacteria. For example, E. coli in this case, it damages the DNA, and once the DNA has been damaged, this bacteria cannot divide into two. This is because the DNA has been damaged so much that proper copies or right copies of DNA cannot be transferred from one generation to another. And that is why the cell decides to go into cell death or apoptosis, and finally the cell dies. Okay, so this is how ultraviolet light works. However, of late, people have realized that E. coli and even certain organisms have something called as a repair mechanism. In this repair mechanism, which is called as the dark phase or the dark repair mechanism, cells repair their DNA, which has been broken down because of UV light, and then they get reactivated and start growing again. So if you are having 100 bacteria or 100 cells of E. coli in your solution, 90 of them are going to die because of the UV light, but 10 of them are going to repair their DNA and then come out of it and start dividing again. So this was a problem. So the whole paper or the whole research focuses on usage of some other chemicals along with ultraviolet light to stop what is called as DNA photoreactivation or DNA dark repair. A small hypothesis has been given on why they are doing this and what they are expecting out of it. They say that combined UV and photocatalytic disinfection, that is ultraviolet light combined with photocatalytic, that is something in the form of a chemical that they will be adding, will decrease the dark repair. The chemical that they are adding is basically titanium dioxide. The concentration will decrease the rate of dark repair in E. coli. And even the accession number and the strain number of the cells, ATCC 10798, everything has been mentioned. So briefly, concisely, they are trying to put in as much information as possible. What are the objectives? To determine the effect of photocatalysis on reactivation on E. coli after UV disinfection in water and study the reactivation kinetics. As we already know that we know about the process of deactivation, we are trying to check whether they get reactivated or not. The methods have been shown pictorially. As I'll again reiterate that as Pranay sir had told, अगर आप इसको basic images में दिखा पाओ, if you can show certain images or show certain steps or a flow chart, it becomes very nice. So what they're trying to do over here is they have introduced a UV lamp, they have introduced a water surface, and then they have grown the cells on a culture. And finally, they have added titanium dioxide to the culture to just check whether the cells, do they regrow, whether the cells die or the cells are perished or not. The results are pictorially represented over here. If an onlooker has time, he can look at the pictorial representations, or they can directly go on to the discussion and then finally to the conclusions. The discussion says that higher titanium dioxide concentration suppresses the dark repair. How does it do it? By stopping the by basically conducting the oxidation of the E. coli cell membrane. By oxidizing the E. coli cell membrane, they are creating holes or punctures inside the cell membrane, and finally, all your external reagents are entering inside the E. coli cell and damaging the cell. The doubling time of E. coli has been increased. So what is happening? For example, if your E. coli takes 18 minutes to double, the best indicator of declining population of E. coli is the increase in doubling time. So if your cells are now taking 22 minutes, 24 minutes, 30 minutes, it means that your cells are now under stress. Most of them have died and the remaining are also going to die. What are the conclusions? They have listed down the conclusions in 3 very good points. 1 gram per liter of titanium dioxide induces photocatalysis, and it results in higher disinfection, log reduction, the same thing that we were speaking of. A substantial amount of the population of E. coli cells has died. Lower dark repair rate. Basically, they were trying to give a hypothesis over here that if you add TiO2 along with ultraviolet light, there will be lesser and lesser amount of reactivation and lesser amount of repairing of DNA taking place. Higher doubling time, this is what we have already discussed. Higher doubling time of the E. coli after UV and photocatalytic disinfection, in contrast to using ultraviolet light alone. Okay, what is the future work? Future work basically यहाँ पे हो सकता है that you can work on this thing on other pathogenic organisms such as bacillus, other gram-positive bacteria, to check whether this is also feasible for other gram-positive organisms, because while you are disinfecting water for drinking purposes, you have n number of organisms. This is the study on only one of them. So further work or future work can be done on this. And finally, the references, which is the most important part. There are two huge references given over here. Basically, they must have referred certain papers, they must have read a lot of articles, and उसमें से उन्होंने methods and objectives निकाले होंगे, and combining all those methods and objectives, they came upon this paper. Okay, so the references have been mentioned. When was the paper published? Where did they work? All of these things are mentioned over here. So this is how a very brief, concise, and a beautiful looking paper looks like. Okay, so if anybody has any questions, or if there is anybody who has not understood, we can talk about it again.
Yeah, anybody have got questions in their mind? Please ask, because this workshop is only for you people, to understand how the research e-poster is to be presented, how it should reach to lot and more people in a convenient and easy way. So you can see here that sir has explained in a very beautiful manner with an example consisting of, so you should by now you should know there are 4 or 5 things which is required in e-poster. That is introduction, objective, abstract can be there, methods, discussion, conclusion, future work, references. All these 4 to 5 topics should cover your e-poster. So e-poster is made up of all of these things. Okay. So anybody has got questions in their mind, they can ask without any hesitation, please. Anybody? Okay, thank you so much, sir, for
Thank you, ma'am.
explaining it in a very clear and very easy way, and with a very good example. Thank you so much, sir. Now I request Priya Borekar ma'am to continue the workshop.
Yes, ma'am.
Yeah. Wait a minute. Wait a minute. I will make you co-host.
Yes.
Yeah, you are co-host now.
Okay, okay,
Yeah.
ma'am. Am I audible to all?
Yes, ma'am, you are.
Okay. My screen is visible to all?
Yes, you can continue.
Okay. Good morning, first of all the students. As our topic is making of e-poster on the research work. Already Anuja ma'am, Pranay sir, and Akash Singh sir give the wonderful presentation on that. And the continuation with the overview of some more examples of e-posters are there. So in introduction of e-poster, the Anuja ma'am already told about the how it is important for the mature students. Okay, but now this is a very simple example for the e-poster. So photosynthesis, everybody knows the photosynthesis, but how to make a presentation of that, or e-posters, that is very important. So in this image you can see very beautifully. That is the photosynthesis given, and the sunlight is absorbed by the prokaryotes, and green pigments when the chloroplast are there. There is the image of your chloroplast, and the reaction is given. So the carbon dioxide plus water gives the glucose and the oxygen, which is already known to you all. But we can give the in a presentation manner. That means e-poster, as already Akash Singh sir told to you that it is very interesting and beautifully. That means it is very important कि जब भी आप कोई पोस्टर देखे तो उसको देखते ही आपको एक व्यू आना चाहिए कि what is exactly happen in the photosynthesis, or what is happen in the reaction, or what is happen in the posters. Okay, so overview, e-poster is nothing but the overview. So this is the some examples. Here the limiting factors are given, what happen in the light intensity, carbon dioxide concentration, and the temperature. There are the different types of graphs are there. So here the if the concentration of light intensity is there, if the concentration of carbon dioxide is increases, these are given very beautifully. That means pictorial graphs are there. So you have to present that your work in a on a single paper. Okay, so this is very easy to save your time in the case of how to present that e-posters in the any conferences. Pranay sir already told you the due to the pandemic situation we can't go here and there in a conferences, and there is the no presentations, and how to improve your skill. It is very important to e-posters. So these are the photosynthesis example. Another is your, just wait. Again this is your next presentation, where the introduction part already Anuja ma'am told to you that the 4 to 5 things are very important. That is introduction, aim, methods, and conclusion, results, and references. So this is the paper having the unraveling the pathogenesis of the citrate diamonosis. Which gives the aims and methods, how the results are given. That is the very important. Again the different types of your graphs are given. These are the different making your mind. That means molecules, motions, and memory is given. That is your new topic, how to memorize and how the your central nervous system is work. So these types of e-posters actually gives the memory. That means the graphical representation or pictorial memory is clear to you. That means it is visible in after a few days also. Okay, so already अनुजा मैम told to you कि जब भी आप कोई पोस्टर ई पोस्टर बनाते हो, that is the easy for the organization also, electronic way to presenting the posters, and it is very permanent. Okay, so if you see the this type of e-posters, that means it make a visual memory to all. That means if you know the how this is the cell body or dendritic axons or myelin sheath are there. So here are the diagrammatic representation or poster. Here in a one paper you know the all about the how neuron works, how using the different types of axon, terminal mycelium sheath, axon, etc, 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 but in a single paper way. That is very important. Again next is your what is DNA. You already knows about the DNA, but if you can see this poster, the you already know about that, but you have to memorize it, or how to prepare that poster. That is very important. In research work also you have to present that poster. So it is very important to how to easy way. That means it is a very convenient and eco-friendly type of poster, because the environmental friendly, that means the sociality and the organization and for the researcher also it is very beautiful. Okay, so this is very easy method, and how to present and how to it is where more advantageous, and it is not carry anywhere. So it is very important to research work or the other organization to carry these e-posters. These are the different benefits of the presentation of the e-posters also. Okay, student, any query about that, you can ask the question. These are the I'm actually taking the very simple topics, because you are now the in a final year, but in a MSc level you can know the how it is the representation. These are the our some examples.
Okay.
And
Anybody has got any questions, please ask. You can ask to any sir, any ma'am. Do you have any questions? Students, you can ask it. Yeah, ma'am, you can continue with vote of thanks.
okay. So students, this is a very nice platform for in a pandemic situation. So I am very much thankful to uh, Mrs. Anuja ma'am, which having the given the introduction of the e-poster in the pandemic situation, and she already told you the how the importance of that e-posters. Actually, you are already matured one. This is why she told you ki, why this program for you people. Okay, so the again the physical contents not be able to present here in the pandemic situation, so we have to carry that material, that means e-poster or electronic way we are presenting the posters. She beautifully shown us give the information about the e-posters. Again, I am very thankful to the Mr. Pranay Sadavarti sir. They are giving the such a wonderful steps and procedure how to make the e-posters in this pandemic situation, and how to very much fortunate about the that means he everybody clear the views about the how to share the idea, how to COVID situation. There are the no avishkar here this year. Again, the particular format is told by the Pranay sir, and the actually you are in a future perspectives in MSc levels. So participation in the conference that is not possible, but how to compelling of that research work, he can say very beautifully. So again, he told about it in a presentation, that is the title, abstract, again the material, methods, discussion, results, conclusion, and the references. These are the very important topics of the e-posters. Again, I am very much thankful to Mr. Akash Singh sir. He wonderfully given the nice presentation, and they are giving the hypothesis of the presentation. That means research work, how it is important, and the reactivation of E. coli of water after the UV photocatalytic disinfection. This paper is also explained by the sir. Already sir told you the how the pictorial presentation is important, and the again the e-poster you have to in a one piece of paper. So graphical representation is very much important, how the society is beneficial, and the organization and the research, all the things told by the Akash Singh sir. So I am lastly conclude my vote of thanks, and I am very much thankful to principal ma'am of Santaji Mahavidyalaya who giving such a opportunity to our department to presenting the making the e-posters in a pandemic situation. So these are the importance and introduction of art. So it is thankful to all students also, they are listening patiently, and it is because of you only people, this program is very successfully. So thank you one and all. Thank you technical persons also, all the persons who are making our program successful. Thank you so much, ma'am. Thank you, sir. Thank you.
Okay. Thank you, ma'am. And I am very much thankful to all the final year students without whom this program is not successful. Now, I request everyone to fill the feedback link. I will share in your group, and all have to compulsorily fill the feedback link form, which will be available in few minutes after this workshop. Okay, now workshop is completed. Thank you so much, students. See you later.